How can studying somatic mosaicism benefit understanding of human health? So the whole reason that we do all of these scientific studies is that we really want to forward our progress of understanding why people get sick and how to better treat them. And so in the SMaHT consortium, right, we're really trying to understand, if we look at all these different tissues in someone's body, right, and the genetic profiles of those different tissues, how can we better understand if someone has a heart condition or a lung condition or a neurological condition, the genetic profiles that are present within those tissues. And so in this consortium, right, we're really trying to go really deep and understand small nuances in genetic profiles. And then we can link that back to human health and human disease states with the ultimate goal of trying to create a more personalized medicine and a healthier society.
One of the key questions in the field of somatic variation is understanding its disease mechanisms. Why do some somatic variation lead to disease while others do not? We still don't know too much about it, but we do know is that mechanism is really context specific. For example, the same mutation can cause disease in one tissue but totally harmless in another. What SMaHT is doing is that it building a complete and comprehensive somatic variation atlas across a variety of human tissues and the development stage and across different biology modalities. This really enables the downstream analysis to trace back a mutation in time and space so that we can pinpoint the exact mechanism and the context where the disease is initiated.
There's a lot that we do not know about the mechanism of human disease, and genetics and particular what we would call traditional genetic studies where you look at inheritance haven't been able to explain all of the molecular underpinnings and all of the architecture of human disease. Now, what's missing? What's missing is an understanding of some of the nuts and bolts and things that can happen that can both give rise to human disease but can also be somehow coded in and causative. So this somatic mutation exploration project now offers us one more lens into what disease mechanisms might be driven by molecular factors.
What makes SMaHT technically difficult or challenging is what I find the most exciting part of this program. So SMaHT is a program in which we're looking for mutations in cells. And if you're driving down the road in New England in the fall, you look at the forest, look at the leaves. When you look at the leaves, they start to change slowly. So over time, the cells in our body, we all start as cells that are semi the same, and they start to change over time. And so I think of it as like driving down the road, where's that one perfect yellow leaf? That's what the scientists in the SMaHT program are going to do. They look at the human body and they need to find the one group of cells, one branch of cells that have mutated, changed. And now let's find those mutations and map them, and then we can apply them to human biology disease. And that's an amazing question, because when you know that, we can apply it and make a resource for the world. But beyond the scientific community of SMaHT, the whole scientific world can look at this and use it to human health and development for going forward. So next time you're in the road, driving down, look at the forest. That's what the scientists need to do. And I want the one perfect red, yellow leaf. And you find it, and that's what they need to do. That's the challenge, that's the excitement.
Somatic mosaicism, where different cells in the same individual carry different genetic information, is changing how we think about aging and development. Traditionally, we assume that all the cells in the body, they share the same genome, but emerging studies and discoveries are showing that somatic mutations can arise throughout life, shaping everything from tissue functions to disease. In development, mosaicism might help explain how cells diversify and specialize, even in the absence of environmental cues, and in aging, accumulating mutations throughout the lifetime, from the time that we are born to the time that we are in our old ages, is acting as a biological clock, revealing the tissue-specific wear and tear, and even early signs of dysfunction before disease arise. By mapping somatic variation at scale across tissues and time points, the SMaHT consortium is helping uncover how our genome evolves within us, and how this shape health across the lifespan.
What excites me most about potential clinical applications of somatic mosaicism? So, there are a lot of people out there who have disease, a rare disease, an undiagnosed disease. They go through a whole diagnostic odyssey, they don't have any answers. The thing about SMaHT is we're shining a light on this sort of hidden genetic variation that's not going to be ever found if you only sequence someone's blood. So the clinical application I'm most excited about is I think we are going to be a first step into making diagnoses for a lot more people who have rare genetic disease, because we are going to show that we have to look at the DNA that is in tissues besides just the normal tissues that we use to sequence blood.
The purpose of building a catalog of somatic variation and the tissues they impact is to better understand human health and disease. Having that catalog helps us to understand what variation impacts various tissues and cells. The types of variation that we see is informative, but in all, it truly helps us to understand health and biology. So by understanding this variation, it enables us to better diagnose, better treat, and hopefully better prevent the disease types.
In my view, the biggest gap in studying somatic mutations is studying them at a single cell level. So cells, they are fundamental blocks of our life and of our bodies. And cells in our bodies, they do different functions, they're located in different regions of the body, and they actually live different lives. So cells in the skin, they're exposed to environment, sunlight, UV damage. Cells in our brain, they do different functions, so they have exposure to different things. Cells in our guts, so they're constantly interacting with food and drinks we have, so they have different exposures. So besides different functions, there are different exposures. So, and that actually eventually reflects into their somatic genome. So cells, genomes of the cells from different organs and from different regions of our bodies, they actually accumulate mutation differently. And so far, we have very limited abilities to study mutations at a single cell level. In the SMaHT consortium, so we have multiple groups which are developing, testing, and applying approaches to studying somatic mutations at a single cell level. So not only technological advantage, the key advantage of SMaHT consortium that we have multiple tissues from donors, from the same donors. So we can study and ask a question, how different mutation accumulate in lung compared to liver, or compared to colon, compared to skin, compared to brain. So in each person we can actually see what was the differences and how the difference may project to the environmental exposure, lifestyle, and other factors contributing to this.
One of the things that makes the study of somatic mosaicism technically challenging is that the variants that we are looking for in the tissue that we're studying are only present in a very small fraction of the cells of that tissue. And so it's kind of like the needle in the haystack analogy, where you have to fish through a whole bunch of hay to find something that looks very much like a piece of hay, a needle, and that would be the cell that has the somatic variant that's of interest in all of these studies that are giving us information about how those somatic variants contribute to human health. And so first there's a problem of finding it. The other technical challenge is the sequencers that we use to spell out the DNA sequence of the tissues we're sequencing are not perfect, and they make mistakes. They make errors, often at a predictable rate. And so when we fish through the hay pile to get to the needle, sometimes we can't tell if that needle is actually a somatic variant, that's a true somatic variant, or it's just an error made by the sequencer. So in the analogy of the hay, is it actually a needle or is it just a pointy piece of hay?
Why is it important to build a comprehensive catalog of the somatic variants? Well, the goal of the network is to identify and characterize somatic mosaicism in human genomes across representation of multiple cohorts. And not only is the network really concentrating on generating the best kind of data that enables scientists to identify somatic mutations, many of which can be quite rare because they can occur later in life and therefore only affecting a fewer number of cells and making it harder to detect by sequencing approach. We are also a network composed of best scientists in the community, in my opinion, and we are trying to push that edge, the frontier of the scientific innovation, so that experimentally and computationally coming up with approaches and best practices that allows us to identify somatic mutations as well from the data that we generate. And so this catalog is supposed to be the culmination of our analysis, where we're putting together basically a reference of what one would expect to find in terms of genetic variations in the normal genome. At the Data Analysis Center, we have built the data portal that will host these sequencing data, as well as the analysis result in a searchable and easy to find and visualize on our data portal. And so this somatic catalog of highly curated, high confidence, many of which might be experimentally also validated, that are also easy to find and easy to understand, so that we're hoping to put it up in the portal with annotations as to whether it's found in the normal population or whether it has some pathological implication, as well as other aspects, not only genetics but epigenetics, transcriptomic information that could be also elucidating what the effect of the somatic variants that we might find in the genome of our cohort.
What exactly somatic mosaicism means in terms of thinking about the concept of personalized medicine. It's first important to think about what exactly is personalized medicine. And really the concept of personalized medicine is taking into account all the information about the individual sitting in front of you to try to tailor their care such that you're best actually caring for that individual, taking all these pieces into consideration. What we know is genetics right now is actually having a big impact in terms of our ability to implement personalized medicine, because we can now understand if someone, say, is at risk for a particular condition later on in their life, we can actually tailor their care in terms of actually maybe even doing preventative screenings for this or changing the medications that we're prescribing to them. Somatic mosaicism impacts this similarly. And this is not necessarily something that you're necessarily born with, but these changes in your DNA that occur throughout your lifetime that can have a direct and dramatic impact in terms of how exactly an individual may experience health as well as disease. There's many examples of somatic mosaicism, both in terms of causing cancer, but also causing different conditions that can result in, say, their immune system being more aggressive in terms of fighting their body than it should be, or causing growths on part of their body. And what we know is that taking these somatic mosaicism features into account has really made dramatic improvements for some of these individuals, even resulting in situations where drugs that have been initially developed for cancer are now able to actually be utilized in individuals who have somatic mosaic variants that are causing impacts in terms of their health and actually letting that impacts be resolved or significantly mitigated. I think one of the major challenges, though, is that we just need to have this map of somatic mosaicism and understand how to precisely understand and map it so that we can actually get to this point where this can be something that's implemented in personalized medicine. And that's what I'm really excited about the SMaHT program for getting us to that point.